Hi everyone, this is K. Ramnachari, a faculty member. Today we are going to discuss about the topic cell biology. The topic name is ultra structure of animal cell. Ultra structure of animal cell. Very important topic. First of all, cell. This is the one of the cell. Cell is the structural functional unit of organism. Cell is the structural functional unit of organism. Totally two type of cells are there. Prokaryotic cell, eukaryotic cell. Prokaryotic cell main important role. A pro means primitive, karyotic means carrier, indicate nucleus. Primitive nucleus, prokaryotes. Example, different type of bacteria, archaebacteria, eubacteria, cyanobacteria, different type of bacteria, mycoplasma also. And after that, eukaryote, U means true, karyotic means nucleus. The clarified nucleus, clearly nucleus. That is the eukaryote. Eukaryotic good example, protozoa. Protozoa is a single cell animal. But number of students protozoa is eukaryotes, prokaryotes. Statement is correct. Some are tell about that answer, but the wrong. Protozoa 100% eukaryotes. Why? Protozoa single cell animal but contain different type of nucleus. There is a protozoa, plants, animals are eukaryotes. But this is the alter structure of animal cell. Different parts are there. Alter structure of animal cell. For example, alter structure of animal cell. Cell first cells are mainly cells two parts. Main cells two parts cells are there. First cell discovered non-living cell, living cell. Non-living cell, living cell. Non-living cell first discovered Robert Hooke, scientist. Living cell discovered first time Anthony van Leeuwenhoek. Different living cell and non-living cell. And first electron microscope, common microscope discovered Anthony van Leeuwenhoek. Compound microscope Jacarius Johnson. Electron microscope Nall and Ruska. Okay, my dear students, today the cell is the structural functional unit of organism. Cell mainly three parts are there. First part, cell first part plasma membrane. Main important cell membrane. This is the plasma membrane. First cell. Second one cytoplasm. Middle structure cytoplasm. Third one is cytoplasm, different type of parts, Golgi apparatus, lysosomes, and after that ribosomes, different parts are there. And final one, nucleus. This is the nucleus. Our body different systems are there. Our body different systems are there. Number one, our body different systems are there. Number one, digestive system, nervous system, and after that circulatory system, respiratory system, different systems are there. The main control system, nervous system. Main control system, nervous system. But the cells different parts are there. Plasma membrane, cytoplasm, nucleus is available. But main part is nucleus. This is the controlled part, nucleus. And after that, this is the model cell. This is the model different type of cells are there. This is a plasma membrane, cytoplasm, nucleus. Okay, next continue. This one is a cell. This is the model cell. So continue cell parts. This one is also anatomy of cell. Detailed anatomy. Actually, plasma membrane, cytoplasm, and nucleus. Plasma membrane and cytoplasm and nucleus. Different cell structures are there. And after that, this is the this one is main important role anatomy. That is the main important plasma membrane. This is the cell three parts. First part. Second one is main important cytoplasm. Cytoplasm different type of rough endoplasmic reticulum, smooth endoplasmic reticulum, peroxisome, lysosomes, microtubules, Golgi apparatus. These are totally cytoplasm. Nucleus, the control of this cell, main important is control nucleolus, nucleus sac, and different type of structures are there. Very important role in a cell structure. Okay. This alter structure of animal cell, plasma membrane. Plasma membrane, this is the cell membrane, is very important role, outer protection role, and permeability, semi-permeability, selective permeability, different specified character, plasma membrane. These different theories are there. Plasma membrane, main outer membrane of the cell that control movement in and out of the cell, double layer phospholipid bilayer. Main important role, plasma membrane. All cells are bounded by a thin membrane, cell plasma membrane. The plasma membrane is also called cytoplasmic membrane. First discovered Kramer and Nageli. And main important role, plasma membrane. Different theories are there. Davison membrane model, lattice model, micellar model, Robertson model, and after that this one is also fluid mosaic model. Next coming video, detailed study plasma membrane. Now this one only outer structure, cell membrane, outer membrane of the cell that control movement in the and out of the cell, double layer phospholipid bilayer. And after that, next one is cytoplasm. Cytoplasm main important role. This actually cell, plasma membrane, nucleus. Between the colloidal part, gel-like mixture, cytoplasm, surrounded by the cell membrane, contains hereditary material. This contains different type of cytoplasm, hereditary material. The translucent heterogeneous colloidal substances enclosed by the plasma membrane is called cytoplasm. Cytoplasm consists of various molecules such as water, salts, sodium, potassium, and organic compounds such as carbohydrate, protein, lipid, nucleic acid, and enzyme. Cytoplasm contains a number of membrane-bound bodies called cell organelles. Different cell organelles also available. Endoplasmic reticulum, Golgi complex, ribosomes, lysosomes, mitochondria, centriosome, and kinetophore microtubules. These are totally organelles. Now first one, endoplasmic reticulum. Endoplasmic, this is the model, endoplasmic reticulum. This is one of the cell organelles. Cell organelles, endoplasmic reticulum, main important role. Endoplasmic reticulum moves material around in cell. This is the endoplasmic reticulum. First time endoplasmic reticulum proposed by Porter scientist. Scientist name is Porter. Smooth type lacks ribosomes. Smooth type. And transport by vesicles, formation and lipid. Synthesis in some cells. Rough type ribosomes embedded in surface. Transport and protein synthesis. Actually, endoplasmic reticulum two types, rough endoplasmic, smooth endoplasmic reticulum, absent of ribosomes, rough endoplasmic reticulum, present of ribosomes. Rough endoplasmic reticulum, it use only synthesis of protein, and smooth endoplasmic reticulum, synthesis of lipids. Now next, continue. Ribosomes. Pillade first described the organelle. They are either attached with the membrane endoplasmic reticulum or salters freely in the cytoplasm. Ribosomes are the sites of protein synthesis, so they are called protein factories. Main important granular structure is the one of the organelles in cell, ribosomes. Protein factories, our body structure, ribosomes. different type of granular structure. For example, endoplasmic reticulum present of ribosomes rough endoplasmic reticulum, absent of ribosomes smooth. Actually, this one is each cell contain thousands embedded of rough endoplasmic reticulum floating throughout to the cell make protein. So this one is the protein factor is our body structure. Next, mitochondria. Mitochondria, the term mitochondria was introduced by Benda but proposed bioflux to Altman and Kolliker discovered mitochondria are semi-autonomous organelles of the cell. They may be cylindrical or spherical shape and occur in large number in metabolically very active cells. Mitochondria are the ready source of energy required of the various microbic activities in the cell. Hence, mitochondria are described the powerhouses of the cell. And mitochondria main first time discovered Kolliker, but bioblast statement Altman, but Benda first time name allotted Benda, but powerhouses of the cell and cell respiration and ATP produced spherical to rod shaped organelles, which is a double membrane powerhouse of the cell, produces energy through chemical reactions breaking down fats and carbohydrates. Controls level of the water and other material in the cell, recycles and decomposes protein, fats and carbohydrates. Next, lysosomes. Lysosomes, the first discovered crystal by Didouet. Lysosomes are membrane bound vesicles. They are filled with hydraulic enzymes. They help in intracellular digestion and extracellular digestion, autolysis. These organelles are described as the suicidal bag of the cell. This one is made suicidal bag of the cell lysosome. Round organelle surrounded by a membrane and containing digestive enzyme. Digestive plant of proteins, fat and carbohydrate transport undigested material to cell membrane for removal. Cell breakdown of lysosomes exploded. Crystal Didouet first discovered. This is the powerhouses of the cell. Peroxisomes. Peroxisomes Peroxisomes also very important role. That's enzyme containing structure that help the detoxify alcohol from bile acid and break down fats. Vacuoles is the plant cell, very important role. Water solution. Main membrane bound sacs for storage, digestion and waste removal. Cytoskeleton and centromeres and microtubules. Centromeres was discovered Van Benden. It is otherwise called cell center. The centromere plant play the important during cell division. The centromere absent in plant cells. Microtubules hollow rods that function primarily to help support and shape the cell. Golgi bodies. It was discovered by Camillo Golgi. It consists of cristae, vesicles and vacuoles. The main function of Golgi complex is secretion. A membrane bound vesicles of placental sacs and sacs parallel to each other. Protein packaging move materials within about the cell. This is the Golgi complex. Golgi bodies. Microfilaments. Microfilaments also important. And nucleus. The nucleus was first observed Robert Brown. Generally, the nucleus first observed Robert Brown. Generally, nucleus is around the situated in the center of the cell. The nucleus is a dynamic center within control of regulators the various metabolic activities in the cell. The nucleus contains nuclear membrane, nuclear sap, and chromatic reticulum nucleolus. This is a chromosome made up of DNA and nuclear membrane surrounded nucleus made up of layers opening hollow material enter in the nucleus and a nuclear membrane the surrounding nucleus made up of two layers opening hollow material enter and leave nucleus and main define of the cell. A cell is the smallest unit of the capable of performable life function vary in both size and form. Category cytology is the study of cell. Okay everyone, thank you so much. This is the today topic main important role alter structure of animal cell. Thank you so much. Do like and subscribe my channel.